Well, first let me welcome you on behalf of the faculty. As a fellow educator, I really welcome the opportunity of being able to speak to you, particularly as a parent as well. My daughter actually just graduated from high school last summer, and she's now just starting her first year at college, but I have a 9-year-old who's going to be following on. I just have one story that might be of interest to you. So I went to a parents' meeting in middle school and talked to her science teacher. And he said, "Your daughter is doing very well in class. By any chance, are either of you scientists?" My wife and I are both scientists, and I made the big mistake of coming home and telling my daughter what the teacher had said. And then the instant identification that she could be read in any way by her parents' interest killed science for my daughter. And then she decided that that was it. So she took no interest after that, I'm afraid. So be careful what you say to parents. I should say, though, I think before that time she had the scientist's logic in how she thinks about problems. And in her interest in international relations, I think that will be helpful. So you'll be aware that there's a great deal of interest and excitement in the area of regenerative biology and its application to regenerative medicine. And what I want to do today is present an argument illustrated by some of our own interests as to how developmental biology provides the rational entry point to this problem. So the science of developmental biology has at its founding goal the determination of the molecular and cellular principles and processes that direct the transformation of a fertilized egg into a new member of a species. And if we look at this from the perspective of a human embryo, we see that the fertilized egg undergoes a number of cleavages over a period of a few days to establish the blastocyst. And then this structure implants into the uterus. And then the real business starts in the process of gastrulation, which is a few days after the period of implantation. And what gastrulation does is it transforms the embryo, which is essentially a single layer of cells, into 3 layers of cells, the so-called primitive germ layers. And these are going to give rise to different parts of the ensuing body plan. So, for example, the ectodermal layer will give rise to the CNS and differentiated cell types of the CNS. The mesodermal germ layer will give rise to structures like the kidney, and the endodermal cell layer will give rise to the pancreas. And these events will elaborate over a protracted period of fetal development as all the different cell types get established for the body plan. So there are several different characteristics of a developmental program. So one obvious characteristic is that there's a large increase in cell number as we establish the cellular template that's going to enable the formation of all the different cell types of the body. As development progresses, we're going to see an increase in cell complexity. So the germ layers are established at gastrulation, but they're still relatively simple populations of cells. Later in development, they're going to give rise to the wide variety of different cell types. So complexity at the cellular level will increase dramatically as developmental time goes on. We can say something about the potential of cells. As the cells become increasingly specialized, their developmental potential becomes increasingly restricted. And this is important. So the cells of here within the blastocyst, these inner cells have the potential to give rise to an entire conceptus. But that's not true of these cells at this later stage. They have much more limited potential to give rise to the different cell types of the body. So as cellular complexity increases, the potentials of cells decreases. The next general property is interactions between cells. So all these events are not intrinsic properties of the cells themselves. It requires the interactions between cells for these events to occur. Notably, it requires signaling events that occur between cells. Further, many organs are a composite of cell types that originate from the interaction amongst the germ layers. For example, if we look at the gut, it has an epithelial population of cells on the outside that's involved in resorption of food, which is derived from the endoderm. But the movement of that food is dependent upon the muscle layers that surround that, which are derived from the mesodermal population of cells. And the last general characteristic of developmental programs is the aspect of pattern. So that all the organ structures are not laid out in a haphazard fashion, but they're laid out in an invariant manner within a species. Although that pattern of organization by which we position the organs in the body may be different from one species to another. So there's something superimposed upon the process by which complexity is generated to ensure that the right cell types form in the right place within the developing embryo. So those 5 events characterize the developmental process that leads to the foundation of an adult body plan. But as we're all aware, development is not over by the time one's formed an adult. So each of the different organ systems of the body has to be able to repair and regenerate within the adult. So when we think about how repair and regeneration occurs within the adult, one paradigm that's come to the fore in recent years is the so-called stem cell paradigm. So in this, there's a limited set of stem cells which for a given tissue has the interesting property that they can renew themselves and then by an asymmetric division give off daughter cells that, for example, can amplify the population and then differentiate into a wide variety of cell types that are necessary for a given tissue or organ. And there are several very good examples where stem cell-based systems maintain organ systems within the adult body plan. So for example, in the skin, there are stem cell populations that maintain the turnover of the skin and generate the various appendages that are associated with the skin, for example, the hairs. And in the gut, there are stem cells that lie within the endodermal compartments, the digestive epithelium that surrounds the gut, that maintain the generation of new cells in the villi. And then in the male gonad, there are spermatogonial cells that maintain the continued production of sperm. And then from the clinical perspective, perhaps the most famous of all are the hematopoietic stem cells of the blood system that maintain the ability to generate the wide variety of blood cell types. And if we look at these stem cell-based systems, we can see that there's a number of characteristics that they share in common. So the tissues in which they operate in are often exposed to the environment and require a system because of the rapid turnover of cells that can rapidly replace those cells. So there's a fast turnover of many of these tissues, although the stem cells that repopulate these tissues actually divide at a slow rate. And so they give off cells that are undifferentiated and amplify as an intermediate stage. If we look at stem cells in these different systems, so for example, if we compare hematopoietic stem cells with the stem cells of the skin or with stem cells in neural systems, we find that there are some shared molecular characteristics, suggesting that there may be some properties that stem cells have in common. But if we ask, can stem cells cross tissue barriers, despite what you may read in the newspapers, I think the general consensus of the scientific community is that stem cells don't readily cross their tissue barriers. So for example, hematopoietic stem cells form new blood cells, but they don't form new neurons.
So basically what you're saying is that different types of stem cells depending on where in the body?
Exactly. So the stem cells may share some properties in common, but the stem cells that are present in the skin will not repair and regenerate muscle. And the stem cells that are in the bone marrow will not form a new neuron. So they're limited in their ability to do that and restricted to the tissues that they normally repopulate.
What about cells in the umbilical cord?
So cells in the umbilical cord fall into a category of stem cell called a mesenchymal stem cell. And this stem cell appears to have properties where it can form a number of the mesodermally derived tissues at an early stage.
So they're capable of crossing barriers.
So those ones represent, if you like, an earlier cell at an earlier time in the embryonic program, for which the strong evidence for a counterpart of those cells still present in the adult body doing the same thing is very minimal. So in thinking about how to rationally approach the problem of regenerative and cell-based mechanisms, I would argue that we should do so informed by the normal processes that construct and maintain the individual organ systems. So that developmental biology is a rational approach to regenerative and cell-based medicine. So what I want to do today is talk about two developmental areas that have interested us, and then discuss how the workings in this area is shaping and defining the use of this knowledge towards therapeutic goals. And I'm going to talk about areas that interest us, but I'm not going to talk exclusively about our own research. So as a disclaimer, I'll say instantly, don't attribute everything I'm going to say to me. I will acknowledge the various people whose work I'm highlighting as very nice examples. So I thought I would just choose two problems that interest us. So one is the problem that deals with forming a particular type of neuron in the CNS, the motor neuron. And then I'll talk about a much more difficult problem, which is the problem about how one deals with regeneration and development of a whole unit of an organ, which is the nephron, which is the functional unit of the kidney. So first, let's just consider the motor neuron. So the motor neurons are present throughout our nervous system, from the midbrain region all the way down through the spinal cord, and they're a ventrally located population of cells. So this is a section through the spinal cord. This is the dorsal part of the spinal cord. This is the ventral part of the spinal cord. And here are the motor neuron cell bodies resident here in the ventral part of the spinal cord, sending out their axons to the targets that they innervate, the muscles whose contraction they're going to control. And their action in this muscle here, in conjunction with the sensing of tone in the muscle by sensory neurons that relay that information there, will, I hope, later on this evening, enable me to get some liquid reward for all my efforts. So we know of a number of diseases where motor neurons are key targets of those diseases. So unfortunately, in children, spinal muscular atrophy leads to the loss of motor neurons and childhood death. And then in an adult population, the loss of motor neurons in amyotrophic lateral sclerosis, or Lou Gehrig's disease, leads to a devastating disease for which there's no cure. So how can we look at this problem of the motor neurons from the developmental biologist perspective? So the first thing that we want to know is, how do you make that cell type during the course of development? How do motor neurons form? So the motor neurons arise during early fetal life. And what you're seeing here are two transverse sections through the spinal cord region, this case of a mouse embryo. And so this would be the dorsal half of that spinal cord, and this is the ventral half. And this represents a mouse embryo at about E10.5 days. And so this is long before any neural circuitry is set up. And we're looking at establishing the progenitor cells that are going to generate the differentiated cell types of the central nervous system. This is mouse. This is a mouse, yeah. This is a mouse at E10.5. I should say that if you were to put a human one in front of me here, you would trick me, because I'm sure I would not be able to tell the difference between the two. So if we look in the ventral neural tube with an antibody that recognizes a transcriptional regulator called OLIG2, we find that the OLIG2 cells are located to a tight cluster here, close to the ventral midline of the neural tube. And OLIG2 is a marker of early motor neuron progenitors, and actually its activity is absolutely required to be able to make motor neurons. So the question is then, how do these motor neuron progenitors form in this particular part of the neural tube? And the answer lies to the left. So in the early 90s, some good colleagues and myself identified a family of signaling factors in mammals, chicks, and fish called the hedgehog family of signals. And one of those, sonic hedgehog, which you see here, the protein from the sonic hedgehog gene distributed throughout its normal signaling domain in the CNS, acts as the principal factor responsible for taking a naive population of neural cells and inducing different types of ventral cell identities. So what's the evidence that supports that statement? So there are several lines of evidence consistent with this idea. So first is that sonic hedgehog is present at the right time and place to have this activity. Second is, if we mutate the sonic hedgehog gene, and we can do this specifically using gene targeting approaches in mouse embryo stem cells, and then introducing those specific genetic modifications back into the mouse germline, and we remove exclusively the function of sonic hedgehog, it leads to the loss of all ventral cell identities. If we do another type of experiment in which we take the gene that encodes sonic hedgehog, and then we make a transgenic mouse that now produces that signal up here, 180 degrees away from where it's normally produced, now it induces ectopic motor neurons in this position. And then, as we dissect the pathway through which this signal acts, we find that there's a requirement for each of the pathway components that are involved in transduction of this machinery for the specification of motor neuron identity. So to summarize, a lot of work by our groups, by the group of Tom Jessel in particular as well, we have this prevailing view now as to how you make different cell identities in the ventral neural tube. And this view has a localized source of sonic hedgehog. Actually, it initiates not from within the neural tube, but from a cell population that underlies it, a rod of mesoderm called the notochord. And from there, the signal arises and forms a diffusion gradient, and cells sense the concentration of this, and at different concentrations activate different transcriptional regulators that will lead to the specification of distinct types of neural progenitor identity. So where there's a low amount of the ligand, that low amount of the ligand specifies ventral interneuron cell identities. Where there's an intermediate level, it leads to the specification of motor neuron identities. And where there's highest levels closest to the source, it leads to the specification of yet distinct cell identities. So in this, sonic hedgehog acts as a morphogen where the concentration that a cell sees dictates the response that that cell makes and the neural identity that forms. So we can summarize motor neuron development from the developmental biologist perspective is that sonic hedgehog is the key inducer of the progenitor population. All motor neurons are born, like all cells in the spinal cord, during fetal life. There's no intrinsic repopulation of these cells at any time thereafter. So can a strategy be developed that could generate functional motor neurons? So we know how they're made. Can we take that knowledge and develop an approach that enables us to make motor neurons? So one approach you might imagine to do this would be to take neural stem cells. You've heard of neural stem cells. There's a vast literature about neural stem cells that are isolated from the central nervous system. And then give those cells sonic hedgehog and see whether you can make a motor neuron. And the answer is you can't. And the reason you can't is that those neural stem cells only represent a very small fraction of the possibilities that are open to an early neural cell during the formation of the nervous system. And they have an extremely limited capacity to make the different cell types of the CNS. So what we need to do is go back in time in some method that better recapitulates how you generate the motor neuron population. And this is one of those points where I will acknowledge that this is not my work, but it's the beautiful work of Henrik Wichterle and Tom Jessel. And so what Wichterle and Jessel simply did was use the logic of how we understood you first made neural tissue and then secondarily patterned it with signals like sonic hedgehog to make motor neurons in a test tube. So in this procedure, they take mouse embryo stem cells, which represent a stage of mouse development long before neural cells form. And then through a very simple procedure that simply aggregates these cells and cultures them in a minimal medium, these make cells that are, if you like, generic early neural cells. In actual fact, it's easier to do this for neurons than for almost any cell type. So although neurons have an exalted status in our view, from a developmental biologist view, they actually are one of the simplest cells to form. So whereas most cell types require positive input to form that cell type, making a generic neuronal progenitor cell type actually requires the absence of information. And so if you remove as much information as possible in terms of different signals, those cells will make neuroprogenitors. And then you can take those neuroprogenitors, and now you can give them sonic hedgehog. And now, just as the cells would normally develop in vivo, they now form the range of different sonic hedgehog-dependent cell types. So amongst this mixture are yellow cells that are motor neurons. And then when you've got the basic assay established that Wigtele and Jessel developed, then colleagues like my colleague here, Lee Rubin, who's very interested in motor neuron-based disease, can twist and tweak this basic assay. So now almost all the cells under a set of conditions can make motor neurons. And now one's in a position that you can make literally hundreds of millions of motor neurons in a very simple procedure in a test tube from embryo stem cells. But are these functional? They express genes that are indicative of a motor neuron progenitor. But as I hope I pointed out, this is a long way before the process of making a motor neuron that has an axon grown out to its target, innervating its target, and regulating muscle contraction. So evidence suggests that this is the case, comes from nice work, again, by Henrik Wigtele and Tom Jessel, in which they took the motor neurons differentiated from mouse embryo stem cells in response to sonic hedgehog, and then they introduced those into the neural tube of a chick embryo. It's just very simple to do this procedure, since you can very easily manipulate the chick embryo, and you can imagine that it's much more difficult to manipulate the mammalian embryo developing in utero. Those cells will integrate into the neural tube of a chick, and then if one compares the trajectory of the normal motor neurons here in orange with the cells that are transplanted, which are labeled yellow, you can see that those motor neurons actually follow the normal tracks and go out and innervate the peripheral target muscles, just as the endogenous neurons do. So the evidence says that these are bona fide motor neurons. So then what can we do with this? So the we here, again, is not myself, but it's two colleagues, Tom Maniatis and Kevin Egan, two colleagues in MCB. So they reasoned that this would be a good way to try and approach the molecular processes that are going on in a mouse model of familial ALS. So as you may know, that Lou Gehrig's disease, ALS, is associated with a component of which is familial and inherited. And amongst that component, there are a few genes that have been identified that are associated with that inheritance. The best studied of those are mutants in an enzyme called superoxide dismutase, which has specific amino acid mutations in the human. If you take the human gene that carries those mutations and introduce it into the mouse, excuse me, to make transgenic mice, those mice are a very faithful recapitulation of the progressive loss of motor neurons that you see in ALS. So what they reasoned was that you could develop ES cells from those mice that carry the SOD1. You could develop motor neurons through this embryo cell to embryoid body to sonic hedgehog differentiation assay. And then you can take those motor neurons and then study the properties of those cells. And when they study those properties in vitro, in a nice work published a little earlier this year, they found the important finding that whereas the presence of this mutated form of SOD1 leads to a poor survival prognosis in motor neurons, it's present in non-motor neurons, the cells that actually support the motor neurons, the glial cells within the neural tube, also has a dramatically detrimental effect on the motor neurons. And so now you have very large quantities of these different cell types that you can generate in these in vitro assays, and new ways in which you can explore the genesis of these diseases. So where does the future of this lie? So clearly some future lies, since one can generate these motor neurons now quantitatively, in having a substrate that one can use for exploring how you might repair using those cells. Since there are human ES cells, a future lies in developing these approaches so that you can get reproducible differentiation of human ES cells to make human motor neurons so that you have the closest possible model to the human condition. Ideally, you would really want to try and recapitulate a model that lies as close as possible to the human condition. And here, the potential development of patient-specific human ES cell lines using somatic cell nuclear transfer-based approaches seem to be the best way in which one could pursue the development of human ES cell lines that provide the best model of what may be going on in this disease. And these are interests that my colleagues Kevin Egan and Tom Maniatis share. But aside from studying these cell types and studying their properties as motor neurons, now with very large quantities of these cells, they themselves can be direct targets for drug-based screens, where one can look at the properties of those cells and ask how those can be affected and hopefully ameliorated by any particular drug. So applying this rational developmental biology approach then enables one to develop new strategies to explore new ways in which one could tackle this disease. So now I want to switch to a second, and in some senses a much more complex process, since this is both complex and actually poorly understood. And this has to do with the kidney and how the kidney functions. So as you know, the primary job of the kidney is to maintain a homeostatic balance of water and salt and to remove nitrogenous waste in the form of urea. And this is accomplished by this workhorse tubular epithelial system of the nephron. And this is capable of filtering all the plasma in our bodies several times over the course of an hour. And so what happens in this process is that plasma filtrate comes from the vascular system into the glomerular end of this network, and then it passes through this tubular system. And in that process, it resorbs everything that one doesn't want to lose and fine-tunes the water-salt balance of the plasma filtrate as it passes through this tubular system. and then what's left passes out through this system, the collecting duct network, off to the ureter and the bladder. So let's take the developmental biologist's approach then, and then ask the simple question, how do we form a nephron? Well, the nephron forms from a simple epithelial vesicle structure called the renal vesicle. And it's this simple structure that gives rise to the whole elaborate tubular network of the nephron with its distinct physiological domains that carry out the activities of the nephron at different positions along the length. So then how does the renal vesicle, the precursor of the nephron, form? So the kidney development starts in the mouse, which is the system that we work with, at E11.5 days of development, when this tube here, which is the tube called the ureteric bud, grows in to an area at the level of the hind limb where these mesenchyme cells have been specified to form the kidney. After it grows in there, what that tubular epithelium does is it starts to grow and branch amongst those mesenchyme cells. And this is a movie from Frank Costantini at the University of Columbia. And so now you see over a period of about 2 days, that epithelium has undergone a large amount of branching. It continues to branch over many, many days, and actually in the case of the human, over many months, and forms a continuously large, elaborate tree of a branching network. And that forms the collecting duct system that's going to be involved in urine transport. But as it branches, it also induces cells that lie above it to form those renal vesicles. It induces loosely dispersed mesenchyme cells above it to form the epithelial renal vesicles, which are the precursors of the nephron. So if we look just 4 days later, so at this time, all we have is a single branch. 4 days later, that epithelium has now branched many times. And each of those epithelial branches has a green cluster of cells that surround it, from which the renal vesicles are going to form. So if I just draw this schematically, it's the interaction of this blue epithelium, the ureteric epithelium, that forms that branching network, with this population of cells that lies above it that's the motor that drives nephrogenesis. So what these green cells do is that they provide signals that control the branching growth of the epithelium. And what the epithelium does is it provides signals that act on some cells within this population to cause those cells to first aggregate and then to transition into this epithelial structure, the renal vesicle. So then, to go back to my question as to how one establishes the renal vesicle, we need to know the answers to a number of questions. So which cells amongst this population of cells are the cells that give rise to the renal vesicle? What's the nature of the signals that come from this other epithelial population to induce this event? How do we control the number of these so that we make the appropriate numbers of nephrons? There's increasing evidence that if you don't have the full complement of nephrons, there's a very highly increased probability that you may be susceptible to kidney disease. And then what do these developmental mechanisms say about the normal process of repair that goes on in the adult? So if we look at one of those tips of the ureteric bud, in yellow we see the epithelium of the collecting duct system, that branching network. And then in green and red, we see antibodies that recognize different transcriptional regulators that are expressed in mutually exclusive cell populations with within those mesenchyme cells from which the renal vesicles arise. So how do we determine which, if any, of these cell types are responsible for making the renal vesicle? So in this experiment, we generate simple genetic tools, and this is work of Akio Kobayashi in the lab, in which he takes the genes that are responsible for these red and green patterns, and then fuses under the control of those genes by gene targeting, again in mouse embryo stem cells, this fusion protein. So this fusion protein is a fusion of a protein that fluoresces green, literally, and a protein Cre, which is a recombinase from bacteria whose job is to cut DNA at a particular recognition sequence. So now we produce this fusion protein under the control of those genes, here under the control of a 6-2 gene, and now we can literally see all those 6-2 cells because they fluoresce green. The activity of this enzyme Cre recombinase, by cutting DNA, can activate the expression of a marker which, when activated now in these cells, marks those cells and all their descendants for the rest of life. And so we can simply trace what does a 6-2 cell give rise to in the developing kidney. And what we find is that the 6-2 cells label in blue all the cells of the nephron and only the nephron. That other mutually exclusive population of cells labels interstitial cells that surround the nephron and cells that regulate the vasculature. And so we can say then that it's the 6-2 population of cells that are the cells that are destined to give rise to the renal vesicle and to the nephron. So 6-2 population demarcates the renal vesicle progenitor pool. It's rapidly downregulated on induction. It's the target of inductive signals. And I won't show you the evidence here, but it's a self-maintaining property. It's a self-maintaining cell population. So that during the course of kidney development, the 6-2 cells differentiate and replace themselves, much like a stem cell lineage. But unlike a stem cell lineage, it has a limited life, because this ends at the period a few days after birth when the full complement of nephrons has been formed. So then what is the inductive signal that acts on that mesenchyme population of cells to induce the renal vesicle? And here we became interested in a signal which is a member of a different family. You heard about the hedgehog family. This is a family called the Wnt family. And the Wnt family represents a family of signaling factors conserved throughout all metazoa. This particular member of the family, Wnt9b, is expressed within the epithelium of that ureteric bud collecting duct network at the outset of kidney development and then throughout the whole period of kidney development. If we then again use gene targeting through mouse embryo stem cells and introduce a mutation in Wnt9b that removes Wnt9b activity but leaves all other activities behind, we now get mice that are born, but you can see that here's a wild-type mouse in which here's the kidney and the adrenal gland. Here's the adrenal gland, and this is the small agenic kidney of a Wnt9b mutant. And it dies of kidney failure shortly after birth. And if we look for any evidence of formation of nephrons, there's no evidence whatsoever of nephron formation. So Wnt9b, and I won't go into all the evidence, is the key inductive signal. And it's both necessary and sufficient to induce nephrogenesis in the kidney mesenchyme. So then what maintains that progenitor population of 6-2 cells such that one can form the complete complement of kidney cells? And the answer here comes from nice work of Michel Self and Guillermo Oliver's group, and it's actually the transcriptional regulator 6-2 itself. So this shows a kidney after a few days when it's undergone about 3 branches. And these are the undifferentiated 6-2 cells that are lying above here. And in a 6-2 mutant, all of those cells form these renal vesicles prematurely. So instead of only a subpopulation of cells committing to make renal vesicles, the whole population does. And as a consequence, one depletes that population of precursors, and kidney development comes to an abrupt, rapid halt, only able to form a few nephrons. And we know that in this process, 6-2 somehow operates within cells to stop those cells responding to Wnt9b. So there's a yin and yang relationship between 6-2's action and that of Wnt9b. 6-2 acting to try and prevent cells from responding to Wnt9b, and Wnt9b trying to act upon 6-2 cells to induce nephrogenesis. So we have a sense of these key regulators. We're building on this to understand how we can, for example, propagate 6-2 cells in culture, and how we can, for the motor neuron, develop inductive systems where we can quantitatively make these precursors for nephrons. But let's switch to the adult and ask, well, okay, how does the developmental program, where we know how you establish a nephron, relate to the process of how you repair nephrons in the adult? So I mentioned that all nephrons are established during fetal or early neonatal life. In the case of the human, they're all established over a period of a few months during fetal life, in which you induce in each kidney about 500,000 nephrons over a protracted period of reiterative branching of this epithelial network and induction. And in the mouse, you make 50,000. And there's no evidence that after that, you can make any more nephrons. We know they all come from a 6-2 cell. If we trace what happens to 6-2 cells, we see that they're lost in conjunction with the cessation of making new nephrons. And of course, we'd like to know what governs that they're lost at that time, how the system knows that it's formed the appropriate numbers of these nephrons. So in the adult, there is no 6-2 population, and there is no new nephrogenesis. So we can imagine that there's 2 ways in which we repair nephrons when they're damaged, as they frequently will be due to different insults. So one model would have that there's a mechanism of tubule self-repair, where the differentiated cells that are within the tubule have the ability to repair themselves on damage. And another group, another camp, suggests that there's a stem cell that lies out of the tubule that comes in an adult-specific type of a process, and it's able to contribute to the tubules, and that this stem cell then effects the necessary repair. So how can we distinguish between those models? So we collaborated here with Ben Humphreys in Joe Bonventri's lab to look at a nice model of kidney damage that they had developed in an adult. And all this does is it simply cuts off the blood supply to the kidney for a short period of time in the mouse. And this leads to a fairly focal, and it's not clear why it's so focal, cell death in this region, the so-called proximal straight tubule of the kidney. And roughly 80% of the cells die here, and then over a period of several days, those cells are repaired, and the damage is restored. So the question is, how is that repaired? So we reasoned that we could do a very simple experiment to distinguish between the out-of-nephron stem cell repair from the endogenous repair by intrarenal tubular epithelial cells. So with the 6-2 mouse strains, we can literally label every nephron red. And then we can injure the nephron in that model so that now we only have 20% of the cells in that region that are left that are red. If those cells repair themselves, then we make a tubule that stays red. If those tubules repair themselves by some other stem cell coming in, for example, from outside of the tubule, now we will get a mixture of red cells and, in this particular model, green, because it labels every cell outside green. In the way that we've done it, they're not green. They're just not labeled. So in other words, dilution of the red population would argue that there's some cell type from outside the nephron that has the property of being able to repair. And so we do that experiment, and genetically we make all the nephrons red, and then perform the ischemia. And what this leads to is that compared to the uninjured kidney, it leads to cells undergoing rapid rates of proliferation in response to repair. And if we ask what cells are undergoing rapid proliferation, they're all cells that are red cells. And we find that the nephrons are restored exclusively by red cells with no dilution from any other cell type. So we think then that in repairing this system, it's tubular epithelial cells that provide the predominant mechanism by which this system is repaired. And this has parallels with the nice work from my colleague Doug Melton, looking at how the beta cell population, that's the critical cell population in type 1 diabetes, repairs, where the prevailing evidence from his group's work suggests that it's self-repair, it's self-division by beta cells that restore the loss of beta cell complement in disease models, and not from some undifferentiated stem cell population. So we can then take this information about how things normally work to then develop approaches that will enhance endogenous repair, or potentially take this information to develop new strategies in which we can work on the normal mechanisms and test out alternative approaches that we can incorporate the developmental program's role in repair and regeneration of adult structures. So I'm end there, and I hope that I've given you some sense of how a developmental biologist, looking at the logic of how things normally work, tries to adopt a logical framework for thinking about these problems.
You had a thing about that you damage the kidney in an embryo mouse and watch it repair.
Actually, this is an adult.
Oh, no, no.
That's in an adult.
Also, that's my question. My question is, are nephrons repair themselves in adults?
That's an adult. Yeah. So this is an adult model that's recapitulating what we think would be the likely process that would be going on in repairing our kidneys as well.
So there is adult repair of nephrons?
So there is adult repair of nephrons. As you can imagine, given the nephron's job and its interface with the plasma, it's seeing a lot of toxic substances, and it's fairly common that it has to repair and regenerate. And so we think that that's likely happening from an intrinsic cell population. And the simplest model, although we don't have all the data that would say this, is that the differentiated cells that are present within the nephron may be the normal template that's used to repair. So it may be very analogous to the beta cell system in the pancreas, where the differentiated beta cells are the cells that repair there.
And is repair in the pancreas as well?
Sorry?
There's repair in the adult pancreas as well?
There is at some low rate, and experimentally you can actually make that a less slow rate, for example, by doing a partial pancreatectomy, where you can remove a good chunk of the pancreas. And in that case, you see that it's the restoration from beta cells. Yeah?
Have you seen gene linkage that has to do with the proteins necessary for signaling the regenerating in the kidney with certain kidney diseases or predisposed kidney diseases?
Yeah, that's a good question. So you know, we have not, and the kidney genetically has lended itself to really two classes of human mutations that have been, it's been an extraordinarily good model for figuring those out. So the one is polycystic kidney diseases. And that has a very obvious phenotype where one makes a hugely enlarged cystic kidneys. The other one is hypertension, where the kidneys control salt. And so if you look at people who are extremely hypertensive, then oftentimes that relates, and it's genetically inherited, that relates to kidney problems. The sorts of things that operate at the developmental angle, they are much more difficult, I would suspect, genetically to try and get a handle on. I said that there's a prevailing view that if people take people who have had susceptibility to kidney diseases and they do things as straightforward as count nephrons, there looks like there's a strong correlation between reduced nephron number and the propensity to get kidney diseases. And I think there's an increasing feeling out there in the medical community that perturbations in the developmental programs may predispose individuals to certain types of diseases. Yeah.
You had mentioned the chronic hedgehog at different concentrations causes different intracellular responses depending on which way they develop into. Do you happen to know what the mechanism would be within the cell that causes?
I'm glad you asked that question because I meant to put a plug in for those people who are going to visit my lab afterwards. So I have a very good postdoc, Steve Vokes, and he's very interested in why, given a certain concentration of hedgehog, do you make one cell type and another concentration another cell type? And what Steve's going to talk to the people who visit the lab about is his work where he's been trying to understand the transcriptional program that different concentrations of hedgehog unleash, and to directly look at the transcriptional regulators that hedgehog regulates to identify their targets. And this shows that different levels of the transcriptional regulators regulate different sorts of targets. So if you like, the level of a signaling is translated into a different transcriptional output that then enables different target genes to be switched on. And you will see his data and see some of the transgenics that he's generated to demonstrate the target genes that he's interested in. And those are some of the primary transcriptional regulators that get active in those cells, and they themselves specify those cells. Yes?
Earlier on when we were talking about motor neurons, you said that you could take mouse cells, put them in a chick, and they grow.
Right.
What happens if you let the chick grow out? Are they functional motor neurons?
So those experiments were not done to my knowledge, and I don't know whether they have been done. So I don't know the answer to that. I think that it's one of those experiments that if it did work, you could draw very strong conclusions. If it didn't work, you couldn't draw very good conclusions. But they can certainly find their peripheral targets.